Hello, and welcome to this Chemistry World webinar. I'm Mariana Kneppers, Chemistry World Science Media Producer. Now on to some introductions. In today's webinar, we'll learn about the fascinating field of forensic taphonomy. We're delighted to have two leading experts in this field joining us for the next hour. First, we will hear from Noemi Procopio. Noemi is a UKRI Future Leaders Fellow and principal investigator of the Omics team at the University of Lancashire. Her lab uses omic sciences to aid in forensic investigations. She's internationally recognized for her pioneering work, including the first legal case worldwide in which microbiome evidence was accepted in a forensic casework. After Noemi, we'll be hearing from Shari Forbes, coming to us live from Canada. Shari is a professor and chair of forensic science at the University of Windsor, Ontario. Her research focuses on soft tissue decomposition and assists police canine units to improve their training protocols for cadaver detection dogs. Her expertise is regularly requested to assist law enforcement with locating and recovering buried or concealed evidence. We will be ending the webinar with a Q&A session with both of our panelists, so please be sure to submit your questions. And finally, given the subject of our webinar, we wanted to give an advisory that these presentations may include some graphic content relating to death that some viewers may find upsetting. These images have been included to show the scientific process in action in this field of study. That being said, viewer discretion is advised. Now that's enough of me talking, all of my chatting. Let's get things started. To begin, we're going to do a short poll to get an idea of who our audience is. There we go. This poll just gives us an idea of who's in our audience today. Now, as Noemi and Shari will explain, forensic taphonomy is a multidisciplinary field, including biology, chemistry, and many other sciences. So this poll will help us to let us know who's out there listening today and set the stage for these presentations. We have some results coming in already. Chemistry naturally being the leader, given that this is Chemistry World. And we've got forensics taking up the lead as well. Well, not the lead, but second place. Gosh, this is exciting. It's like a horse race. Okay, it looks like we've got a slowdown in responses. I think it's safe to say that we have a majority of chemistry viewers in the audience, followed by forensics, and then biology with a few others in the background. So without further ado, I'll go ahead and pass over to Noemi to give her section of presentation.
So thanks everyone for being here today. It's a pleasure to be here discussing about forensic taphonomy with you all. The title of today's webinar is Forensic Taphonomy, the Chemistry of Delivering Justice. And specifically for this half part of the webinar, we're going to talk about the forensomics approach. Forensomics is the name of the group that I'm leading and also the research that I've been doing since 2019, thanks to my UKRI FLF fellowship. I'm based at the University of Lancashire, and without further ado, let's start to delve into post-mortem interval. So for those of you not familiar with post-mortem interval, when we refer to post-mortem interval or time since death, we are essentially considering the time between death and the recovery of a body. Depending on how much time passed, we can have more or less decomposed remains. When we are starting at looking at a fresh animal, in this case, this is a small piglet. To give you some context, this piglet was placed on the soil, as you can see, on the grass in England in June. So relatively, I would say, cold environment, despite being summer, but we know how summer is in England for those of you living here. After 14 days, the piglet was in this condition. As you can see from this picture, we have an active decomposition stage, and we mean active decomposition when we have, for example, fluids getting outside the body, these dark decomposition fluids that I'm sure you can see all around the pig. Part of the head was already more in an advanced decomposition stage where the maggots were actually feeding onto the carcass. Maggots and flies are actually more active, particularly starting from natural orifices in the body. So it's quite peculiar to see face base and other points of ruptures being the ones that are more colonized by maggots, and therefore those losing the most of the mass in a shorter time frame. And as you can see here, there are also lots of eggs being laid down by insects coming on the carcass. After 7 days more, actually, the result was this one. So in England, we could see that basically in 21 days, we just had some skeletal remains and a bit of fur there. When we have different decomposition stages, we can somehow infer the time elapsed since death, especially when we have a fresh body. We can look at, for example, the body temperature or the insect colonization, which is something very important to give us some information about the minimum time elapsed since death. However, when we are in circumstances like the last one, or even better, in conditions like this one, where we have bones versus bones, it's very challenging to understand for how long they've been there for. And it's basically almost impossible with the methods that we have right now to be able to discriminate the time elapsed since death. Especially in these two cases, you will see bones have a different color, but it's not necessarily associated with the time elapsed since death. It may be that the body on the right-hand side here has been exposed to more sun rays, for example, which are causing bleaching of the bones. So it's very challenging to understand something about post-mortem interval in these scenarios. Besides bones in general, decomposition is affected by a lot of extrinsic and intrinsic variables. When we talk about extrinsic variables, we are referring to any variables which is not directly affecting the body, but it's taking place outside the body. For example, the temperature to which the body is exposed, the humidity, cycles of wet and dry, or exposure to sun rays, also the type of soil in which the body is placed, especially if the body has been buried, and also the action of scavengers, which are those animals feeding onto the body. On the other side, we also have a series of intrinsic variables which are able to affect how fast or slow decomposition proceeds. Some of them being, for example, the body mass index of the person, if the person was an active person or more sedentary type of person, the age of the person, the cause of death. We know that if a person is dying because of a natural cause, decomposition may be slower than someone dying by violent causes with a lot of injuries on the bodies, because there will be points of access for insects, as we discussed a bit earlier. It's also important, the intake of drugs and medications and antibiotics, because they can slow down the progress of decomposition, because they are affecting on one side our internal microbiome if they are antibiotics, or they can repulse the interest of insects if they are drugs, for example. So also in this case, we can see differences in the pace of decomposition. So our idea was to apply omics technologies to improve post-mortem interval estimation. When we are referring to omics technologies, we are referring to the study of a full set of specific molecules in a tissue or in a full organism. So if we are talking about proteomics, for example, we are talking about studying the full set of proteins in that tissue or individual. If we're talking about metabolomics, it's the same at a metabolite level. If we're talking about transcriptomics, it will be mRNAs essentially. So in addition to these main omics, which are constituting the central dogma of biology, I also want to include here also the study of metagenomics, which is the study of the bacteria surrounding us and within our bodies as well. So metagenomics is referred to the study of the microbial populations present within and around our bodies. All of them, as we will see today, can help somehow into improving our ways to estimate post-mortem interval. And in particular, as you can see here, highlighted by these arrows, transcriptomics, proteomics, lipidomics, metabolomics, and microbiome can all of them be useful to estimate and to get some extra information about post-mortem interval. As you can see, the applications in forensics is not just associated with post-mortem interval. We can also get some more insights, for example, on cause of death or age of death. We're going to touch briefly on age of death estimation using proteins as well. Despite today's focus is going to be mainly on PMI. Let's start with proteomics, which is, I would say, my first love, because my PhD was in the application of proteomics to forensic science. The assumptions for using proteomics for PMI is that in vivo aging and post-mortem taphonomic processes of biological tissues can have an impact on the proteins that we can recover from the body. So on one side, we can expect a reduction in the proteome complexity. So due to leaching phenomena, if you imagine all of these balls representing different proteins with similar colors being similar proteins, you will see that with the passing of time and the decomposition of tissues, you may expect some of these proteins to undergo degradation over time, and therefore, when analyzed with a mass spec, you will just see less of them and also a smaller variety. On the other side, we can expect an increase of post-translational protein modifications, PTMs, especially those that are non-enzymatic modifications, therefore not needing a living being to take place. Some of them are, for example, deamidations. So deamidations can take place naturally on proteins, and we can see an accumulation of deamidations represented with these different patterns on some of these proteins over time with increasing post-mortem intervals. This is the type of sampling that we conduct. Normally, we are working with bones, and now more recently, we also started to focus on, I would say, easier tissues, such as soft tissues and biological fluids. But bones are hard, not as a mean of fact, but they are hard to be worked with, and we need specific protocols to work with them. As you can see, sometimes we are doing minimal invasive samplings, which consists in using a small Dremel bead and getting some bone powder, creating these minimal invasive lines that can give us enough material to do proteomics. Some other times we do more big, I would say, cuts. This one is a window cut on a human tibia, and then we put the bone chip into a freezer mill, which is performing a conversion into powder using liquid nitrogen. So this is minimizing the degradation that could be caused by heating, for example, something that may happen instead when we use Dremel. And then the bone has to be decalcified using an acid. So the acid is decalcifying the hydroxyapatite mineral, which is composing the bone, and releasing the proteins in solution. Then proteins are solubilized using some detergents, and then they are loaded onto some special filters. In this case, this is an S-trap device, which is consisting in a filter which contains a C18 membrane, similar to an HPLC column, that can trap the proteins and then remove all the detergents, which will, of course, affect the mass spec analysis. We don't want any detergents in our samples. And then proteins are digested into peptides using, for example, trypsin, and then they are eluted from these filters, and they are concentrated and purified, ready to be injected into the HPLC MSMS. The instruments that we use normally are instruments such as an Orbitrap, and we obtain mass spectra, essentially. So the first paper I want to quickly mention you is this one, because this is the first one that we did working on human-donated bodies instead of animals, and that has a special place in my heart. I particularly love this cover art because this is actually the rendering of one of the 4 donors included in the study, which I find really nice. As you can see, in this case, we were sampling both the iliac crest and the tibia of these 4 donors, and we wanted to see if there were correlations between the proteins and, on one side, the age at death, so the chronological age when these people died, and the post-mortem interval. As you can see here from this highlight, there are some proteins which are significantly correlated with the age at death. In this case, we have increasing age and a decreasing relative abundance of phytoin-A protein in bones. And regarding PMI, for example, this protein, which is a collagen protein, is decreasing constantly with increasing time since death. Of course, this one was just the first pilot study that we did. It's just 4 donors. Now we expanded our studies to about 60, 70 donors, and we are currently looking for highlighting the best biomarkers for both age of death and post-mortem interval, starting from a tiny little amount of bone, which is about like 25 milligrams. Second aspect of our research is using metabolomics and lipidomics for post-mortem interval. This is a very similar concept. We are still starting from a tibia sample, for example, or another dense bone. And we can use different methods to extract either polar compounds, so small molecules, metabolites, or lipids. The way in which we like doing these different analyses is to actually use two separate extractions. We use the chloroform methanol method, adding also water to separate lipids from the rest. And we actually use this extraction to identify and analyze lipids. And then we tend to use methanol acetonitrile water to extract metabolites from the same samples. Also in this case, samples are run onto either a GCMS or an LC-MS instrument, such as a QTOF, for example. This is a study that it's currently under review. Hopefully it's going to be published very soon. And also in this case, we are talking about bones. As you can see, this is the mandible. This is actually a pig mandible that we cut into 4 quadrants. We had many mandibles taken from the butcher, and we placed each of these quadrants at a different either depth, from 0 to 50 centimeters, or we left them for different times, so from 1 to 6 months. The idea here was to try to understand if the burial depth has an effect on the metabolomic profile of these bones, and also if we can still find some biomarkers useful to estimate post-mortem interval starting from bones on a shorter time scale, which is between 0 and 6 months. The results were pretty good. We're quite happy with them. As you can see, there is a distinct cluster between the control samples, so the month 0. This side of the plots are samples run with the GC instrument, and this side is run with the HPLC instrument. It's very similar, though. You can see that the samples at time 0 are separated from the others, and then they are almost following a pattern following the increased time since burial. And when we were doing a PLS modeling, we were able to estimate the post-mortem interval, or we should say post-burial interval, with an error of, especially if we look at this one, 0.48 months, which means about 15 days over 6 months time period, which is something, again, that we could never do just by looking at how the bones look like. So we are quite happy with these results. Last but not least, probably and arguably my favorite, is the use of microbiome for post-mortem interval estimation. This is based on the assumption that microbes live within our bodies and on our bodies, and post-mortem they start to follow a specific succession, which we can partially know in advance, and by using that succession, we can estimate a post-mortem interval. To do so, we need to decide which approach we want to take, either a targeted approach when we look at specifically, for example, bacteria. In this case, the approach will be called amplicon sequencing. We will target a gene which is called 16S rRNA gene, and that one will be amplified and sequenced, and that will give us an idea about which bacteria are present in the sample. Or we can take a metagenomics approach, which is full, is not targeted at all. In this way, we can look at all DNA present, so not just bacteria, but bacteria, eukaryotes, fungi, viruses, and others. The first idea of the microbial clock was published in 2016 by Metcalf Group, and as you can see, we can expect a series of different decomposers, bacteria represented in blue, eukaryotes, non-fungal represented in yellow, and fungi represented in red, which are following specific trends, starting from a fresh kind of condition and body, going to active decay, advanced decay, and finally to dry remains. So that's the assumptions on which the use of microbiome is important for PMI estimation. In this case, I'm going to show you a study that we did in extreme conditions. This was done in North Dakota together with a collaborator of mine. As you can see here, the weather in North Dakota is quite extreme. Unfortunately, we don't have anything like this in England, despite I love snow. In this case, we were looking at 3 pigs, As you can see, the temperatures were never going above 0, as you can also see from the pictures. The time elapsed since death of these pigs was between 0 and 26 weeks, which is about 6 months and a half. And we took swabs from inside and outside the nose of these pigs, totaling 414 samples. So this is a completely non-invasive sampling technique that can also be proposed in the future for caseworks, because it's literally a swab. Despite these pigs, as you can imagine, were not decomposing. It's almost like putting a piece of meat in your freezer, luckily, and hopefully it won't decompose. So from a morphological point of view, it will be impossible to determine for how long the pigs have been there for. We were able to get very, very good estimations using microbes. So this is random forest modeling of the post-mortem interval. And this is similar to the plot I showed you before. You can see that with the time that is passing, you can almost follow on the plot a succession in the microbial composition of these samples. And the most important thing to notice is that when we combine samples taken inside and outside the nostrils, and we combine that with the depth of snow recorded and the temperature outside, we can get an error which is about 9.52 days over 6 months and a half post-mortem interval, which is, again, in my opinion, is quite exciting, particularly considering the limits that are normally associated with estimating post-mortem interval in conditions like this one. So the ultimate goal is to perform multi-omics integration. This is the direction that we are taking in our team. So we are combining all the different omics together, trying to understand which one works better in different time points, if they are working better combined, or if they are working better on their own. So some take-home messages for you. Different omics approaches can be applied in specific conditions. And we are becoming more and more aware of the fact that proteins and lipids, which are more resilient, may be better to estimate PMI on the longer time scale. So I would say maybe from 6 months onwards or something like that. Whereas something like metabolites can be useful on the shorter time scales, whereas microbes are working pretty much in any case and in any circumstance. The sources of samples can be various. It's not just bones. Despite we focus mainly on bones today, anything can work, literally a swab on the skin, ocular fluids, muscle tissue fluids, soil, internal organs, and literally everything. If we consider that actually a crime scene, it's potentially giving us a lot of different sources, we may be bold and just use different, because all of them can actually give us some information when we look at the molecular content of it. Instruments to be used are mainly mass spectrometers and sequencers. As we said, mass spectrometers for things like proteomics, metabolomics, lipidomics, sequencers mainly for genomics and metagenomics approaches. And last but not least, post-mortem interval estimation can be improved. So I'm hoping to see an integration of these new methods, which are reported here for you in red, more and more frequently, because this is I guess this is the future, and we just need to be open to this. So I hope that these are going to be starting to be applied more and more, and also being researched more by my colleagues. So this was the last slide. I'd like to thank my team. I'm never able to get a picture with all of us together, so I always need to do a bit of a collage, but this is my team right now, and I'd like to thank them all for the work that they are doing. And I'd like to thank you all for the attention, all my funders, UKRI, NIJ, Science and Technology and Policing, and the British Academy, and a special thank to the body donors and their next of kin, whose extremely valuable gift made this research possible. Thank you very much.
Wow, thank you so much, Noemi. That is fascinating. As a biologist myself, that's my background. I really found the microbial work really, really interesting. So yeah, incredible job. Thank you so much. I'll go ahead and now pass the control over to Shari for her section of the webinar. Thank you so much.
Okay, thanks so much for the introduction, and thanks to Noemi for an excellent preview of forensic taphonomy and human decomposition. I'm actually, this is also my expertise, forensic taphonomy and human decomposition. I come at this from the chemistry aspect, and today I'm going to focus on one very specific aspect in terms of how we can apply this knowledge to canines. And specifically, what we try and do as chemists is understand what human remains detection dogs already know. And you'll see what I mean as we go through this. So for those who aren't familiar with this specialized scent detection dog, human remains detection dogs are really trained to locate a diverse range of human remains. So they could be looking for skeletal remains, such as a perished missing person or hiker. They could be looking for burnt remains, such as those found following a fatal fire. I'm sure you know we use them extensively in mass disasters to look for recently deceased and or decomposed victims. And we can use them to try and locate concealed victims, such as a homicide victim that may be buried in a clandestine grave. So how does the chemistry actually work in this context? Well, as the body decomposes, and you've heard a lot is happening from both the biological and chemical aspect. But one of the major things that's occurring is decomposition odour is being produced. And that decomposition odour comprises hundreds and hundreds of volatile organic compounds. Now, we hypothesize, although we don't yet know, that the dogs are actually just using a handful of these compounds to locate human remains. In other words, they recognize that a few of these compounds get them their reward during training and gets them their reward when they apply this at a crime scene or a scenario. Now, the question for us is, what are those handful of compounds that we actually need to know? The way we study this is at human decomposition facilities. This is an image of our REST facility, currently the only facility in Canada. And obviously we have to use human cadavers to study human decomposition odour. So when we're ready to study this odour, we take one of our donors, usually many of our donors, and we collect an odour sample every day, if not more frequently, such as every hour. When we're ready to do that, we remove the cage that protects our donor, and we place this very large and unwieldy hood over the top of the donor so that we can trap all those gases and volatile organic compounds under that hood. After a period of time, we can then attach, and I apologize, I didn't put on my, I won't worry about it. I was looking for my highlighter here, but anyway, hopefully you can see my cursor. So we just attach a sorbent tube to an opening in that hood, and we use an environmental air sampling pump to draw all of those gases or those volatile organic compounds onto that sorbent tube so that we can then chemically analyze that. Now this shows you a schematic of how that occurs. So you can see our donor is on the surface with the hood over the top. Those gases are then trapped on that sorbent tube, and the air sampling pump is used to draw them on. But if we think about it, when a dog is scenting, they're actually scenting both the air and the soil. And the soil can be very important because this is where decomposition fluid also leaches, and volatile organic compounds can also be retained in that soil, even if the remains have been removed or scavenged or scattered. So we can apply this same technique using a soil probe with a sorbent tube, and again collecting the VOCs of both the soil and the air. When we're ready to analyze these sorbent tubes, we take them back to our forensic chemistry laboratory, and we currently use comprehensive two-dimensional gas chromatography coupled with time-of-flight mass spectrometry to analyze these samples. Now, the reason we use this technique over the more traditional one-dimensional GCMS is that it does have an increased peak capacity. In other words, it means we can increase the number of peaks that we identify. And given that I mentioned there's hundreds and hundreds, in excess of 500 VOCs that we need to identify, this is really important. Additionally, it gives us increased selectivity and sensitivity, and giving the variations of the concentrations that occur during the decomposition stages, that is also very important too. Now, of course, we use the chemistry and the chemical instrumentation to try and understand what, as I mentioned, the biological detection, that is our dogs, already knows. So every time we collect a sample using our chemical instrumentation, we pair that with the biological response so that the dogs can, although they can't talk, eventually tell us what it is they're actually detecting. So most of the research we do is aimed at actually improving their training through enhanced training aids. And as you can imagine, most handlers don't have the opportunity to go to a human decomposition facility frequently to expose their dogs to the different stages of decomposition. So as a result, they tend to use a range of different training aids that they can gain access to. Now, those training aids could include blood, whether that be fresh or degraded. It could be grave soil from a crime scene. It could be clothing from a decomposed victim, maybe scent pads that are placed on a body in the morgue. There's a whole range of commercial pseudoscents which we absolutely do not recommend be used. There's things such as human versus animal tissue, but the ones that we tend to focus on are those that come straight from the human cadaver, such as decomposition fluid from the soft tissue and bone, which is the hard tissue. So some of the questions that we try and address, and I can't talk through them all today, but one of the questions, or many of the questions we get from the handlers, is one type of sample sufficient? So if they just train on blood, can they expect their dog to still find human cadavers? And the answer, not surprisingly, is no. So then we ask, well, then if we had all of these training aids, do they mimic the complete odour profile of a human cadaver, or is there one that better mimics it over the others? And I'll show you some of those results shortly. They also ask how long can they detect the scent. So many of our handlers are actually searching for missing persons who have been missing for years, if not decades. The likelihood of still having soft tissue at that point is minimal, and it's more likely that they're searching for fragmentary bone that's extremely weathered. And the question becomes, is there even detectable odour for our dogs to locate these remains? So that's something we also work on. Sometimes it's just really practical logistical questions. How do we store the samples? As handlers, they have these access to training aids, but should they be keeping them at room temperature, such as they would experience in a real scenario? Should they put them in the fridge to extend their longevity? What about the freezer? So we focus on these questions as well. And the holy grail for us, and one that I probably won't answer in my lifetime, but what are they actually detecting? As I said, the dogs already know what they're detecting. They simply can't talk to us, so we have to use our chemistry to try and understand. So today I'd just like to highlight two of our projects that have provided some meaningful results or impactful results to our handlers, one of which focused on decomposition fluid and the other which focused on bone. The first was conducted by one of my PhD students when she was in Australia. She's now in Canada, looking at whether or not human decomposition fluid from soft tissue as it degraded is a valid training aid for HRD dogs. So in order to do this, we took our decomposition fluid from one of our human decomposition facilities, and you can see it on the left. It's that very dark viscous product. But what she wanted to test was something realistic, and she knew that in a real scenario, the dogs are not looking for this really concentrated fluid. They're looking for something that's been exposed to the environment, including precipitation over time. So she chose to dilute these samples to better represent the concentration of odour that she might be, or that the dogs might be detecting in the field. And so she diluted these down to 10 to the minus 12, just to give you an idea. She also wanted to address that question of how long can we store these samples. So the handlers are known for storing samples for many, many years, because once you have a training aid, you just want to keep it. So she decided she'd age these for up to 2 years, because this was a realistic time frame of how long the handlers may hold on to these decomposition fluids. So we're going to jump straight to the results. On the left is our chemical detection analysis and results. On the right is our biological detection results, in other words, the dogs. Now, I know this is a bit complex, and I'll just talk through what I can. On the left here, you can see these are our 6-month aged samples. And even as we dilute the samples, you can see that the chemical odour profile is changing, because these classes of compounds are changing as well. So in the stock solutions, we have a lot of sulfur-containing compounds. Not surprisingly, these are the ones even the human nose can detect. But as we dilute those, we start to lose them, and we're seeing a lot more of the green, which are our hydrocarbons, and other different classes in there. So we can see that dilution definitely changes the odour profile. Aging probably changes it as well. The question was, do the dogs care? Or more importantly, is it an impact on the dogs, or can they generalize across this and find those key VOCs that they recognize as decomposition odour? And the answer was they can. You can see 100% of the dogs tested, 100% of the solutions, and every time we tested them, we're getting this correct. In other words, they were saying that this is decomposition odour, regardless of what our chemical odour profile was telling us. So that was fantastic. We did this for 9-month age samples and 12-month age samples with the same results, and we thought this is perfect, until we got to the 15-month age samples. And you can see on the left that the odour continues to change. Again, with degradation, it just keeps changing the chemical odour profile. Now we're seeing a lot more alcohols in that red colour up the top. The issue, though, was our dogs were not giving us the same response. Sometimes only 50% of the dogs would detect it, sometimes none of them would detect it, and it was very variable. They could detect low concentrations more so than the higher concentrations, which wasn't making any sense. We thought at this point we'd probably hit the limit of how long we should age, and we were going to make the recommendation you shouldn't age beyond or store beyond 12 months. Thankfully, my PhD student had committed to the 2 years of study, and she'd already started aging the samples, and she said, let's just keep exposing the dogs to all of these samples and see what happens. And it's lucky that we did. Once we got to the 21-month age samples, you can see actually the chemical odour profile starts to kind of plateau a little bit. So it's becoming a bit more consistent, probably because it's at its limit of aging. And the dogs actually started to improve. We started to get more of that 100% accuracy and a bit more consistency in terms of their responses. Now, I can't show you the 24-month samples. I don't have them here. But for our perspective, the most important thing here is not what the chemical odour profile is doing. It's what the dogs are telling us. And the dogs have to reach a certification level of more than 90% accuracy to make sure they're certified in terms of being able to be deployed in real scenarios. And you can see here, these are just 3 of the teams we've plotted, but they were seeing, with the exception of that unusual 15-month sample, they were getting over 90% accuracy every single time. And this is what we want to see. The dogs are telling us, whilst we still don't know what those VOCs are, they are there and they can generalize across that odour. So our recommendation was actually that decomposition fluid is a valid training aid as long as it's stored appropriately and we understand that time frame. They can, with repeat exposure, detect it up to 24 months, but if you're starting to see inconsistencies, we'd probably say 12 months might be the limit. So that's the first study that I wanted to highlight, and that's from our soft tissue. The second study that I wanted to highlight is another conducted by one of my current PhD students, but she did this work as part of her master's. And that's to look at the other kind of tissue that's probably most likely similar, which is bones or hard tissue. Now, we're very fortunate in Canada that many of our police certified HRD dogs are able to train on an ethically sourced training aid, which are amputated limbs. These are limbs that are donated by a live donor at the time of dissection, usually a diabetic patient. And they're provided to the police so that they can decompose these for a period of time, usually about 1 year, before using them as storing them and using them as training aids. Now that's fantastic, but the question becomes, when the dogs are searching, they're not searching for amputated limbs. They're more likely searching for fragmented or weathered skeletal remains, those that have been exposed to the environment for many, many years. So we needed to confirm, is that training aid a suitable replication of what they're searching for in the field? So to do that, we took skeletal remains from the rest facility in Quebec. They had been decomposing for more than 2 years, and during that entire time, they'd been in an outdoor exposed environment, so clear weathering had occurred. Again, we'll jump straight to the results and look at the VOC profile. This is of the amputated limbs, and this just shows you the classes of compounds that we detected. We referred to these as wet bones because even though they had been decomposed for a year, there was still the presence of some small adhering soft tissue, and we could see clear evidence of lipids actually seeping in or seeping out of the bones maybe, but there was evidence of lipid residue on the bones. Even to our human nose, there was a strong odour, and this dictated or demonstrated that there was minimal bone weathering even after a year of outdoor decomposition. Not surprisingly, we detected 859 VOCs, and we detected some of the very common odorous compounds such as dimethyl disulfide, which is commonly reported usually for soft tissue decomposition. Now, how did this compare to our human skeletal remains? Interestingly, the classes of compounds were all very similar. We refer to these as our dry bones because there was no soft tissue, and to our human nose, there was minimal or no odour. As I mentioned, clear bone weathering had occurred. Not surprisingly, much fewer compounds detected. So we only detected 315 VOCs, and we weren't getting those same primary sulfurous compounds. We were getting other compounds that were the most frequently detected. So if we do the comparison side by side, you can see the classes of compounds are somewhat similar, the ratios are different. But what was more different or noticeably different was the abundance of those VOCs. On the left are the VOCs of our skeletal remains, and they're substantially reduced compared to the abundance of VOCs on the right, which were our odorous or training aids, the amputated limbs. And what did this mean from the biological detection point of view? Well, when we compared individual compounds, we found that there were 37 similar VOCs between the two training aids, telling us once again, maybe these are the key VOCs that the dogs are detecting. But once we applied it to our biological detection, not surprisingly, our HRD dogs were able to rapidly detect those amputated limbs every single time, but they found it much more challenging to detect the skeletal remains, at least in terms of their first exposure. It took many, many times for them to recognize that as the odor, and it did require repeat exposure over time and rewarding even on just a change of behavior for them to understand that this was the odor. But like with decomposition odor, with repeat exposure, decomposition fluid, with repeat exposure, we were able to increase their sensitivity. And ultimately, they can now correlate that detection of the odor from skeletal remains with the detection of the odor of the amputated limbs that they use in their training aids and their training scenarios. So from this, we were able to conclude that HRD dogs should be trained on a range of different post-mortem ages of bone. The amputated limbs are fantastic in terms of being ethically sourced, but having those exposure to skeletal remains also ethically sourced from a human decomposition facility just increases their sensitivity and ensures they're covering that full decomposition spectrum of odors that they're likely to encounter when deployed. So ultimately, and this is just to conclude my presentation, is 2 snapshots of things we do for our HRD dogs, but everything we do is about improving their training by really recommending training aids that are going to enhance their success. And we want to do that not only for our current dogs, such as Kai here on the left, and she's part of our Calgary Police Service unit, but also for all the future HRD dogs that are going to come through the ranks. And in closure, I'd like to thank also our human donors, both the living donors who donate amputated limbs and our deceased donors who donate to the human decomposition facilities. I also have to thank all of our certified police HRD dog teams that contribute to this research, and of course my team, who are the ones that actually do this research that we can publish and impact HRD dog handlers across the world.
Wow, thank you so much, Shari. That is fantastic. What an interesting presentation, and always great to have some photos of some puppies at the end. Definitely warms your heart a bit. It must be so lovely to be able to work with dogs on a daily basis. Absolutely love that. So moving forward, we'll go into our Q&A session now. So if Noemi, if you want to come back on screen, we have, wow, so many interesting questions. We'll have to see which ones we should start with. My gosh. Here's one that is really interesting. I would never have thought this would even, I mean, logically, it would make sense that this takes apart. But Simon has asked, and I think this might apply more to Noemi, but Shari, I think you could maybe also answer in terms of volatile organic compounds. But if a person has a bacterial infection when they die, would this have a significant effect on decomposition post-mortem?
Yes, I can start to answer, and then Shari, if you want to comment. I don't know if that will affect also dogs, to be honest. That will be interesting. So from our side, yes, that's important. As I said, cause of death, it's something that can affect decomposition rate quite a lot. The we need to consider that the gut microbiome is the first one that is leading the decomposition, and at least is starting the decomposition. So gut microbiome is normally anaerobic, and when a body dies, the body will convert into an anaerobic environment, which is perfect for bacteria from the gut to colonize, the rest of the body. So of course, if we have an altered microbial composition because of a disease or an infection, whatever. And on the other side, if we have less bacteria because of an antibiotics intake, for example, that will affect the rate of decomposition because that all starts from those bacteria.
Wow, fascinating. And Shari, would that influence volatile organic compounds at all?
It's a very good question because it is in the sort of perimortem and soon after death period. And that's still something we're studying. One of my recent PhD students, currently my postdoc, just published the work on when does the odour transition from live to deceased. And it's a really good question because it dictates which dog we're going to use in terms of whether we're searching for a live victim or a deceased victim. So I would hazard a guess that it probably does impact the VOC profile, but only in that very early, transition period before it starts to determine profile as a decomposition odor.
I see. Okay. That's an interesting note as well. I didn't realize that you'd have different dogs, working off of kind of live, or the more kind of, what's the word I'm looking for? I don't want to say fresh. That sounds quite, but it's the earlier on kind of phases. And then you have dogs that train on different segments. That is really interesting. A follow-up to that, if a person were taking antibiotics, it would delay decomposition. Would that preserve organs? Would that change the time frame for viability?
Yes, it will delay decomposition. Again, every body is different. You cannot generalize decomposition. Sometimes things that are not expected can happen. But in general, we can notice that when antibiotics are taken, the microbial composition is completely altered and it's drastically reduced. So that will probably cause a delay in the start of decomposition. What we also notice is that sometimes it's not just an antibiotics, but it's a drug intake in general, like also other medicines, because they are acting as a repellent for insects. So insects are not really interested in bodies being, plenty of drugs or other medications. So this is also causing a delay in the decomposition because, as I said, it starts from the gut, but then insects start playing a huge and key role in decomposition. So yes.
Oh, super, super interesting. A question for you, Shari, comes from John. Relating to your study that was done in North Dakota, are there similar decomposition studies from tropical and or desert environments?
So I think Noemi spoke about the North Dakota, but I can comment regardless.
Oh, so sorry, yes.
That's okay. I've studied in both Australia and Canada, so I think I've covered both extremes. We definitely have, the environment is a major impactor of decomposition, and everyone will tell you that we really need to know what's happening in our local environment to be able to assist our local law enforcement. So there are many, many studies across the world using animal carcasses or animal remains, fewer studies with human remains because there are a limited number of human decomposition facilities, but we are constantly trying to expand that so that we can have an understanding of the differences between, such as a tropical environment, a cold climate like North Dakota or Canada, or, somewhere in between, something around the equator like Hawaii. There's lots of great studies out of Hawaii as well. And they all vary in terms of the decomposition rate. And of course, that's going to impact our ability to estimate post-mortem interval.
Of course, yeah. Noemi, did you also want to speak to that at all, since it was from your presentation? Apologies for that.
No, that was actually perfect, and I know that Shari has a lot of expertise working in cold climates as well. So yeah, absolutely that.
Wow. Okay. A question definitely for Shari this time. What breeds of dogs would make the best HRD dogs? Are there specific breeds that you work with?
It's a good question because everybody wants the best HRD dog. There's no specific breed, but there tends to be more common breeds that we see. So in North America, we tend to use Labradors, and that's a lot to do with temperament as well as agility. In the UK and Australia, they use English Springer Spaniels. So again, a difference in terms of temperament and agility. There's not really one that has a better sensitivity in terms of their olfactory capability, although obviously those with a shorter nose maybe don't have as many olfactory receptors as those with a longer nose. But no, to answer the question, there's no specific breed, but the most common would definitely be Labradors and Springer Spaniels.
Oh, I see. It's interesting because you see a lot of, almost in all the old-timey films of people running away from the police or something, they always use bloodhounds. Are bloodhounds ever used in this field?
Bloodhounds are used for tracking human scent, so usually looking for live victims, whether that be a victim or an offender. But again, still not as commonly used now. Certainly for live scent tracking, it tends to be more Shepherds or Belgian Malinois that are used. Again, that's just a kind of decision by most law enforcement agencies as to which ones they'll use, and because sometimes they're being used for cross-trained purposes such as crowd control and other things that require something a little bit bigger than a Springer Spaniel.
Fair enough, yeah. Here's another interesting, kind of a theoretical one, but do cadaver imitating flowers also imitate the change in chemistry over time? I'm not sure. I mean, maybe you both have something to speak to that. Feel free.
So I think the corpse flower is what they're referring to, which is a fascinating flower. And there's been chemistry studies done. It doesn't really kind of mimic the different decomposition stages, I guess. What it mimics is, well, the time we can smell it is about the same time that we would smell what we call active decay, as Noemi discussed. And this is, again, because of those sulfurous compounds, so dimethyl disulfide, dimethyl trisulfide. The ones we associate with soft tissue decomposition are the ones being produced by the corpse flower. To the best of my knowledge, I haven't personally done a study. So, yes, they represent one particular stage of decomposition, I would think.
I see. Wow. Noemi, anything to add? No, okay. Fair enough. That is so interesting, though. I actually, I went to a botanical garden recently and had the lovely opportunity of smelling one of those flowers, and it is quite a memorable experience, needless to say. We're also getting a couple questions about careers. We have something from Caitlin that says, how did each of you get into this specific field of work? Was there something that happened in your upbringing, or, is there something that particularly draws you to this field?
Who's going to start? Should I go for it?
Yeah, go for it.
It's a mix of interests and opportunities, I would say, because I always wanted to do forensic sciences, but not necessarily decomposition, let's say. When I started my PhD, that was focused on proteomics and on cadavers because, well, at that stage I was using animal proxies, but still I was exposed to, studying bones and therefore decomposition in general. And I realized I was enjoying the challenge of finding something new for improving these methods to estimate PMI. So I would say I always had the interest in forensics. Decomposition just came in the way when I was doing my PhD, and I found it quite fascinating as a topic.
Oh wow, lovely. Shari, how did you get involved?
Yeah, I mean, for me, it was a love of science. I'm old compared to Naomi, so I'm pre-CSI days. There wasn't CSI when I was studying, so I just loved science. I used to read Agatha Christie, so I liked crime novels. It's pre-Patricia Cornwell even. And I think I just compared those two interests and decided to study forensic science. But the same in terms of Naomi, in terms of opportunity, I had the opportunity to study decomposition in my undergraduate degree, loved what I was doing and just pursued that as a career.
Wow, It's amazing how we get involved into these, such niche fields, isn't it? It's some of my favorite questions I like to ask people. Another question, kind of relating to the kind of day-to-day aspect, I suppose. What do you look forward most in your job? What are the good things you experience? And, I can imagine be quite a heavy field to work in sometimes, but what is it that you enjoy and look forward to in your job every day? Shari, do you want to go first for this one? Just switch the order up.
It's an easy one for me. I get to work with dogs, so a dog will make my day every single day. Even if I'm having a bad day, they can make me happy again. But so I'm fortunate that I get to work with probably a range of people and animals, I would say. I get to teach students who have that same interest that we had for this career, but then I get to work with police who are having such a societal and world impact in terms of what they do. And then I get to collaborate with people like Noemi and other great scientists and do research, which is the other thing we love. So it's just the best of all worlds, I would say, but dogs are probably top of my list.
Yeah, I can't beat that. Wow. I'm a bit jealous.
Yeah, I don't have dogs, so I will just second the list without the dogs, unfortunately. Also for me, it's a combination of, coming to work every day, enjoying being in the lab with all my team and students and postdocs and whatever. Everyone's super, thrilled when we get a new result or, something good. And then I love traveling, and this work is allowing me to travel a lot to meet collaborators such as Shari and many others. So I'm really fortunate for that. So I'm combining a lot of research with, it's a combination of lab research and field work, which I love in first place. It's not really boring. Plus, it's a lot of travels to conferences and, all over the world to take samples. So it's really good. And the fact that we don't have a human taphonomy facility in England is forcing me to go to the States and other amazing places. So I'm actually not complaining too much, despite having one here would be great. But yeah, that's what we have.
Oh, lovely. Fair enough. I think we may have time for one more question. So we have one here from Ada. She asks, do you see any use for AI in your research? You can think about it.
It's a difficult one. I would say maybe not necessarily. Well, depends what we are considering AI. We use machine learning. We do models. So we are, using machine learning to identify features, which may be the future target for targeted assays or targeted approaches for the police and others. So in that sense, yes, because the amount of the complexity of the omics data, it's huge, and we couldn't do it without machine learning methods and computers. Besides that, I'm not sure about, using AI to generate knowledge. It's more about helping us with the analysis, I would say.
Yeah, I can imagine there's so many different factors you're looking at once. It just helps to have machine learning involved to sort out all of the different, potential, I don't know, how it all mixes up together. I'm lacking the vocabulary. But Shari, how about you? Have you used AI in your research? Are you planning on it?
I haven't. I have heard mention of it or suggestions that it could be used for post-mortem interval estimation. And again, I can see how it would be valuable. Like Noemi said, the sheer volume of data that we have, if you could kind of plug in the parameters of a missing person search, and it could tell you this is what you can expect in terms of decomposition at that PMI, that would be phenomenal. And even if it could estimate PMI long term, that's kind of, again, another holy grail that we all work on. I think it's a long way off, but longer term, if that could happen, I could see it being extremely valuable.
Wow, yeah, fair enough. It does seem that at the moment, it just seems to be more data analysis, but there is, there could be potential for expanding on it in the future. Well, thank you both so much. This has been such an amazing hour. Really, really fascinating. I'm just going to send some thank yous before we pop off. Thank you as well to our Chemistry World digital team here behind the scenes, Chris Pink and Ben Valsler, who've been working behind the scenes to make sure everything runs smoothly. I also would love to thank Rupali Dabas, one of our writers here at Chemistry World, whose excellent feature titled What Happens to Our Bodies After We Die inspired this webinar. And a final reminder that you'll get a link to the recorded version of this webinar along with your certificate of attendance in an email later this week. Thank you all for joining us. We hope to see you in another Chemistry World webinar soon. I'm Mariana Kneppers, Chemistry World Science Media Producer. Goodbye and see you next time.